proteins and amino acids. Today we will learn about production of bile pigments. At the end of this class, the student shall be able to define bile pigments, recall important bile pigments, explain formation of biliverdin and bilirubin, explain conjugation of bilirubin, explain formation and excretion of urobilin, explain analysis of urine to diagnose jaundice. So bile pigments are breakdown products of the blood pigment hemoglobin that are excreted in bile. So the two most important bile pigments are biliverdin, which is green in color, and bilirubin, which is orange or yellow in color. So 1 gram of hemoglobin on degradation finally yields about 35 milligram of bilirubin. So approximately around 250 to 350 milligram of bilirubin produced per day in human adult. The term bile pigment is used for bilirubin and its derivatives. So hemoglobin is broken down into heme and its protein part globin. So protein is further broken down into its amino acids, which are reused, and heme is further broken down into porphyrins and iron. Iron is further oxidized into its ferric iron form, and porphyrins is further broken down into biliverdin. Biliverdin is oxidized to bilirubin. So here heme, by the action of heme oxygenase 1, it is converted to biliverdin. So heme oxygenase utilizing NADPH and oxygen cleaves the methynyl bridges between two pyrrole rings. So here it will break the methynyl bridge, and this converts heme to biliverdin. So heme is oxidized by the action of heme oxygenase and results in the formation of biliverdin and ferric iron and carbon monoxide. So here the ferrous iron is oxidized to ferric form. So higher levels of heme promotes the activity of this enzyme. That is, here higher level of heme will promote the breakdown of heme. So biliverdin is excreted in birds and amphibia, and in mammals it is further degraded. Okay, so here further biliverdin is reduced, that is by the action of biliverdin reductase to bilirubin. So that is yellow in color. Okay, so here hydrogen is added to this biliverdin. Okay, so biliverdin reductase reduces one or more methynyl bridge of biliverdin to methynyl group to form bilirubin, so the yellow colored pigment. Okay. So conjugation of bilirubin. Bilirubin is insoluble in water. The protein bound form is transported to liver via circulation. So in hepatocyte it forms bilirubin diglucuronide. Okay, so here the bilirubin will react with the UDP gluconic acid. By the action of UDP gluconic acid transferase, it will form bilirubin monoglucuronide, and again it will combine one more molecule of UDP gluconic acid, so and it will form bilirubin diglucuronide. Okay. Further, bilirubin diglucuronide is actively transported against a concentration gradient. That is, it requires energy for the transport into the bile duct. So the energy dependent rate limiting step is susceptible to impairment in liver disease. Okay. So unconjugated bilirubin is normally not excreted, only conjugated bilirubin is excreted. So bilirubin diglucuronide is hydrolyzed and reduced by bacteria in the gut to yield urobilinogen. It is converted to urobilinogen, so which is a colorless compound. Okay. Most of the urobilinogens of the feces are oxidized by intestinal bacteria stercobilin. Okay. Then the stercobilin gives stools their characteristic brown color. Okay. Further, formation and excretion of urobilin. Some of the urobilinogen is reabsorbed from the gut into portal blood and transported to the kidney. So here it is converted to the yellow urobilin and excreted, giving urine its characteristic color. Okay. In summary, so here The hemoglobin is broken down into globin and heme. So by the action of heme oxygenase, it is converted into biliverdin, which is green in color, and this biliverdin is reduced to bilirubin, which is yellow in color. So this is water insoluble and unconjugated, and the protein bound form of bilirubin is carried to the liver, and so here it is made water soluble by conjugation. So it is converted to bilirubin diglucuronide, and it is transported to intestine via bile duct. So where the intestinal bacteria converts this bilirubin to urobilinogen, and this is converted to stercobilin and excreted in feces. Okay. And sometimes what happens is some of this urobilinogen is reabsorbed, and it is excreted in papilla.